Hello, this is Donna from the Cholangiocarcinoma Foundation. Welcome everyone today. Our speaker today is Dr. Gregory Lesinski, and the topic is uncovering the immune landscape to enhance therapeutic options in cholangiocarcinoma. Dr. Azad, are you on? I believe that you
are. I am on. Can you hear me?
Yes, yes I can. Go ahead.
Okay, wonderful. Should we give it a couple more minutes or should we go ahead and get started? I guess we'll go ahead and get started. We've got 20 people, so. Well, it's my great pleasure to be able to introduce my friend and my colleague, Dr. Greg Lesinski, as the inaugural speaker for this new seminar series that we are doing through ICRN. Greg is a scientist presently based out of Emory University, and he runs a translational laboratory really focusing on understanding the tumor immune microenvironment of GI cancers. And he has had an interest in cholangiocarcinoma for many years, which is how we got to know each other. He did his initial training at the Medical College of Ohio, where he received his PhD, and then moved over to the Ohio State University, where he did his early facultyhood, moving up the ranks there, eventually becoming the associate chair for translational research before he was recruited away to Emory, and now runs the translational GI malignancy program at Emory. Greg is a very well-regarded immunologist. He has multiple R01s that are really looking at exploring the tumor microenvironment, and especially interest in how we can move these basic discoveries into patients. And so he works with many collaborators to move things into clinical trials for patients. And we're really excited to get the value of his expertise during this talk today. So thank you, Greg, for starting with this seminar series today.
All right, thank you so much, Nilo, for the introduction. I appreciate it, and to everyone else for the opportunity to present today. As mentioned, we're going to be discussing immunotherapy and cholangiocarcinoma, in particular the general immune landscape of the tumor, as well as some new combination therapeutics that are in the pipeline or ongoing. So I have a few disclosures just related to off-label use, some consulting and research funding to support our clinical trial correlative studies. We are part of a very robust program of research at Winship Cancer Institute of Emory University. This is a slide that just illustrates the many faces of our GI oncology program that I co-lead with Dr. Basil L. Reyes up here in the left-hand corner. And really, we have a good cast of individual clinicians and scientists from various departments across the university and within our clinical enterprise, all focused on a common goal of integrating findings from the research laboratory into the clinic and back again. So it's really a pleasure to work with this group, and we're growing, it's dynamic, and it's been a lot of fun. I've been here for just under 4 years. My own laboratory is very interested in cancer immunology, and our overall goal is to study those interactions that occur between cancer and the immune system. What we're really trying to do is identify pathways that are shared within tumor cells, as well as immune modulators that we can target, either as single agents or combination approaches to enhance anti-tumor immune responses. And our priority is really these things that we can move into the clinic in what I call the near term. That's really a 1 to 3 year period. So we're by our nature very impatient. We want to make a difference. We want to get things move forward, and we want this beautiful picture I stole from cancerresearch.org to get the T cells to recognize the tumors and eliminate them. So for my talk today, I'll do really 3 parts. The first is a brief introduction, just to bring everyone up to speed who may not live in this space on applying immunotherapy to cholangiocarcinoma. Second, I'll talk about a variety of data, excuse me, that points to the role of cytokines as potent immune modulators in the setting of this aggressive disease. And then finally, I'll finish off with some early results from a clinical trial combining MEK inhibitors and PD-L1 inhibition as a novel immunotherapeutic strategy, and sort of illustrate the various translational ideas that are moving forward. So the idea of cholangiocarcinoma as an aggressive disease is of no secret to this group. In the United States alone, there are usually over more than 8,000 new cases. This number's rising. And other parts of the world, this incidence is even much higher. In particular areas of East Asia and Thailand, the incidence is very high. We know that the disease is driven by chronic inflammatory processes, and there are some environmental factors such as liver fluke infection that do contribute to the incidence of this disease in many cases. The other interesting characteristic of cholangiocarcinoma is its strong genetic component. There are frequent alterations in a number of pathways, many of which we've uncovered and some which we have yet to discover. Some of the ones we know about and we've done some work with ourselves are those involved in cancer cell viability and proliferation. So a number of targets, and certainly the idea of genetic heterogeneity that is really driving the path forward for this disease as a therapeutic approach. So anatomically, many of you, of course, know that cholangiocarcinoma has a heterogeneous appearance based on where it arises in the bile ducts. It can be in the upper portion amidst the liver, such as intrahepatic, or then collectively hilar and distal, or known as extrahepatic. And by and large, all of these presentations of disease are very aggressive. They have poor 5-year survivals, and maybe the extrahepatic has a little advantage, but by and large, we have a lot of work to do in improving outcomes for this disease. One thing that our laboratory focuses on very heavily is this idea and really certainty that cancers are very cellularly complex. They are heterogeneous. They have a number of different cells that make them up. Tumors are not just balls of proliferating cancer cells. They have certainly a number of cells as supporting cast in this whole process. Among those are cancer-associated fibroblasts, certainly endothelial cells forming blood vessels, and then of extreme interest to our group and many others is this idea that tumors have a very dominant presence of different immune cells within them. This is true not only in primary tumors, but also these cellular interactions that are occurring in the tumor contribute to the invasive process, tumor progression, and then certainly many of these same populations, including fibroblasts and immune cells, appear in the tumor metastases. So this is a very interesting interplay that occurs across multiple stages of diseases, including cholangiocarcinoma. And in the immune therapy space, we like to try to think of things in a little more simplistic fashion, and this slide tries to do justice to that. We're really looking at why in your body you have a tumor, and why does your immune system not recognize it and eliminate it as foreign? Well, we like to think of this very simplistically as a balance that's occurring. There's an ongoing balance and battle and shift in the types of immune factors that are present both systemically and in the tumor microenvironment. What we do know is that when the immune system wins on the left side here, tumors are often predominated by Th1 type cytokine skewing. Conversely, that shifts over to the Th2 when the tumor cell wins this battle. And we know that certain cells that are cytotoxic in nature, like NK cells, T cells, and many others, can elicit direct effects that eliminate antigen-specific or antigen-non-specific manners towards tumor elimination. And on the flip side of that, you have other pro-inflammatory cytokines that foster and facilitate expansion of immune suppressive cells like tumor-associated macrophages, MDSCs, or myeloid-derived suppressor cells that we'll talk more about, and many others. So why does this balance occur, and why does it continually flux? And we like to think of things in two categories. The first is those factors that are intrinsic inside the T cell itself that facilitate immune suppression and prevent it from having an ability to eliminate these tumors. So things like PD-1 on the cell surface are this automatic shutoff switch that's upregulated, inhibiting tumor immunity. Parallel to that are robust expansions of many of these immune suppressor cell populations that occur. This is also true in cholangiocarcinoma, and it's really these things and the cytokines that fuel these processes which make up the balance and which way the scale will flip. And we'll talk much more about that. So zooming in a little bit, just to, again, bring everyone up to speed, You've probably all heard and seen commercials and the lay television advertisements on PD-1. These have had dramatic effects, these antibodies, in other types of cancer. They've had very minimal effect, unfortunately, across a majority of GI tumors, including cholangio. But when they do work, this is the foundation of it. So if you take a basic T cell reaction, T cells recognize and eliminate antigen specifically, tumors or pathogen infected other cells of the body. So a T cell needs to see its antigen via its T cell receptor. So the antigen is shown in green. It's presented via MHC molecule on the surface of either a tumor or an antigen presenting cell. That gives the first licensing or signal for the T cell to mediate recognition. But that alone isn't enough. You need a second signal that's afforded by what we call co-stimulation. In this case, CD28 expression on the T cell can bind its ligand CD80 or 86. When this process happens in the right cytokine environment, you will get the T cell activation, you will get proliferation, cytokine production, and a robust cytotoxic immune response that in many cases can hold tumors under control or maybe even completely eliminate them. Well, we've learned from studies with chronic infection using a variety of viral models such as LCMV by people all over the globe, including those like Rafi Ahmed here at Emory, as well as in the setting of cancer, where we have persistent antigenic stimulation. These T cells get tired, they get exhausted. And as a result, they have intrinsically wired machinery that gets upregulated on their surface to shut them off. And this is really done in an attempt to control immune responses so that your host body doesn't over-respond to things it shouldn't. And what happens is PD-1 is upregulated on the surface of T cells. It engages its ligands like PD-L1 or PD-L2, and then this T cell gets shut off and exhausted. It's essentially turned down. So a variety of drugs are out there that can interfere with these interactions, the antibodies, and then they can restore this effector T cell function and in many cases elicit responses against tumors. So this is how the whole system works, and in the immune checkpoint world, one of the things we're really trying to go after. The truth is, there is not just one single interaction, as you all know. PD-1, PD-L1 is one single negative regulatory interaction that's relevant to T cell biology. There are, of course, agonistic interactions with the plus sign in green here. When these receptor ligand pairs get together, it makes the T cell go. It has the opposite effect and invigorates them. And it's really the balance of all these different things that occur within the cell-cell interactions that dictate what's happening at this interface. And not surprisingly, cytokines influence the expression of many of these receptors and ligands. And this has led to a really blossoming pharmaceutical effort across the globe to identify antibodies or small molecules even that interfere with these immune checkpoints in multiple ways. One other aspect of immune suppression, or so we say in cancer, is this idea that there are cells that can feed back and limit overactive T cell or other types of immune cell responses. And I mentioned them earlier, but one of those cell types is shown here. It's a myeloid-derived suppressor cell, or MDSC. This is a really nice figure from Susan Ostrand-Rosenberg, published in an article several years back, but it does a good job illustrating the diverse ways in which this heterogeneous population of immature myeloid cells can interfere with anti-tumor immune responses. It can act directly on T cells, promoting their apoptosis. It can deprive them of amino acids like cysteine that they need for viability and homeostasis. They can certainly expand through secretion of other cytokines like TGF-beta, T regulatory in other cells, and certainly it has effects in limiting dendritic cell maturation or NK cell killing. So these are really big culprits in immune suppression and the failed response to immune therapy across cancers, and may arguably play a particularly relevant role in cholangiocarcinoma. So to us, they represent a very important target, and I'll show you data from our groups as well as others that really speak to this. So how do we better apply immunotherapy in cholangio or biliary tract cancers in general? The reality is that although FDA approvals have emerged quite frequently for other solid tumors, both in Europe and United States. There have been very few outside of genetically defined tumors that have microsatellite instability or other factors that will make a dent in cholangiocarcinoma. So we, again, have much work to do in uncovering what is locking up the efficacy of these therapies and why can't we extend it to this aggressive tumor. So our group thinks about the cholangiotumors like this. We know inflammation is a key factor driving both the progression and the carcinogenesis of these tumors in general. One other characteristic of a cholangiocarcinoma is its intense stromal reaction. Not that unsimilar to pancreatic cancers are the fact that cholangiocarcinomas have an abundant population of cancer-associated fibroblasts or stellate cells that produce an abundant cytokine milieu that then can feed back to promote this suppressor cell expansion and function and limit T cell recognition against these tumors. So with that said, the trend has been in combination therapies. And this is really where our group is going, as well as many others. We're trying to find better ways essentially to invigorate the T cells while we go after many of these other factors. And one of the things that's been tried is combining immune oncology agents, or IO, with various small molecule pathway inhibitors. Some of the genetic drivers in cholangiocarcinoma, FGF, IDH, a number of other signaling pathways that are relevant are listed here, where there's active immune therapy trials either coming down the pipe or ongoing. Certainly, immune checkpoint blockade is front and center across a number of tumors. And then there's been some very interesting data over the years with cell therapy approaches, which I would argue could be a very big thing if we can appropriately apply them to cholangiocarcinoma. In addition, extreme interest is going into targeting these myeloid, macrophage, and MDSC components, depending on which terminology you like the best. Going after this cell population, given its abundance in these tumors, may be a very important way to go forward. And then finally, of interest to our group is targeting cytokines. Cytokines arguably regulate all these processes very intimately. They can expand the myeloid cells, they can come from the stroma and the tumor, they can directly feed back on the T cells and other innate immune cells to prevent their optimal function. So there's a variety of different ways we can go about the problem. But one thing we have to keep in mind is that our understanding of these dominant mechanisms of immune suppression in cholangiocarcinoma are far from mature. We have a lot more basic science work to do to really sort out the details, and hopefully we can do that. So with that said, as a background, we have a number of priority strategies. The first is we want to understand mechanisms so we can inform logically immune therapy approaches. Personalized therapy is also playing a role in cholangiocarcinoma. This has obviously been made evident with FGFR and IDH and small molecule inhibitors. Can we do this with immune therapy? That is the question. It goes without saying, if we can intervene earlier in the pre-surgical setting, we could have greater impact. And then finally, we can't ignore chemotherapy, which is standard of care. Do we combine immune drugs with it? Does prior exposure to chemotherapy impact the immune system in a way that we might choose one immune approach over the other? All these questions are not particularly well answered at this point in time. And I would argue that in order to answer these questions and come up with the best trials, we have to collaborate. So folks in the lab like myself have to talk to clinicians like Nilo and many others. And by doing so, we can bounce ideas off of one another, get down to the real problems, and then ideally shift the paradigm for immunotherapy in this disease. So now we'll get into some data in the second part. We'll talk about the role of cytokines as potent immune modulators in immune response in the setting of cholangiocarcinoma. And really our overall hypothesis is this, is that if we can target key pathways that orchestrate this so-called immune suppression, we could better leverage immunotherapy in cholangiocarcinoma. And our laboratory is fortunate to have a panel of cell lines that are human in origin, many of which are derived from intra or extracellular or extrahepatic or intrahepatic cholangiocarcinoma. We have one gallbladder cancer cell line, and we've taken these cells and just simply looked in the supernatants at what they produce in terms of cytokines in culture. And we see a few trends emerge. The first is that these human cholangiocarcinoma cell lines make abundant levels of colony stimulating factors and other cytokines that have capability to act upon macrophages and these myeloid cell components of the tumors. The second thing that emerged to us that was quite interesting was high levels of IL-6 across most cell lines examined. And we know that IL-6 is an interesting cytokine in its ability to enhance the growth and the survival of cholangiocarcinoma cells. And then these cytokines and others on this list are quite potent regulators of both MDSC differentiation and function, sort of getting at this idea that this myeloid cell predominance is present in these tumors. So IL-6 has an interesting profile when it comes to signaling pathways. It can signal via a classical pathway shown here, where the ligand binds a membrane-bound receptor. This then interacts with molecules aside of it that are also transmembrane, GP130, which happens to be pretty ubiquitously expressed across most cells in the body. So GP130 provides the anchoring for the downstream signaling moieties that get this cascade of phosphorylation events. So by IL-6 binding its receptor and these downstream factors occurring, you get activation of JAK-STAT signaling, MAP kinase signaling, and NF-kappa B, all these canonical inflammatory pathways. IL-6 is also interesting in that cells who don't have this membrane-bound receptor can still respond. Now remember, they all express GP130, but there does exist this very nifty little soluble IL-6 receptor that can complex with IL-6 in solution, and then this can then go onto cells and signal what we call intrans to transmit signals to cells through these same pathways that normally wouldn't directly respond to IL-6. So this makes it a very dominant cytokine in a physiologic system, and one that certainly plays a role. So we did the very simple thing, and this is a paper that just came out a few weeks ago from our team, my graduate student Brandon in British Journal of Cancer. We showed that there's crosstalk that has capability of occurring. If you take the supernatants from the human cell lines and simply apply them to PBMCs or white blood cells from healthy donors, you can activate these same pathways. So IL-6 from cholangio cells can feed back and activate many of these inflammatory pathways in the immune system. Interestingly, we can neutralize out IL-6 with an antibody and stop this whole process from occurring. With regard to GM-CSF, you see a similar signaling cascade, ultimately ending in phosphorylation of STAT5, a different STAT protein. And using a similar strategy, this experiment, if you put human cholangio supernatants on these immune cells, you drive these pathways, and then this can be neutralized away with antibodies. So this signaling from the cancer to the immune system is very dominant and very interesting, and certainly driven by these and likely other cytokines. The next thing we did, we took white blood cells and we put supernatants or positive control cytokines on them, and we did flow cytometry and showed, looking on the y-axis here at phenotypic markers, that you can get MDSC expansion. So you can take essentially a normal cell and convert it to that with a suppressive phenotype. So in red, we have our positive control. This is IL-6 and GM, recombinant cytokines alone. You get a little increase in the cells phenotypically defined as MDSCs. But when you add these cancer cell supernatants, for the most part, you get a significant increase in vitro MDSC expansion. We, of course, have to confirm that these cells are functionally suppressive in order for us to call them MDSCs for immunology terms. And to do that, we do a very simple thing. Same thing, supernatants from the cancer on the immune cells. You differentiate them into your myeloid cells and isolate them out with beads, while at the same time, from the same autologous donor, we get T cells, we label them with a dye, and then we activate these co-cultures with beads that provide that critical signal 1 and signal 2 to the T cell. So this causes them to proliferate. As they proliferate, they lose the dye, and we can measure that by flow. So these guys right here, this bar, are T cells proliferating alone without any myeloid cells present. And as you can see, they do just well. But over here, if we add myeloid cells differentiated with the cytokines, you reduce the T cell proliferation, and then with the supernatant from the cholangio cells, it has a similar effect. again implying this cancer immune crosstalk makes them functionally suppressive. So work from other groups, including a really great paper published out of the RISV group at Mayo just, I think, a few weeks ago, showed that MDSC are certainly a dominant force in immunotherapy resistance in cholangiocarcinoma. So their group showed very interestingly that tumor-associated macrophages, rather than the cancer, are actually the primary source of this PD-L1, the ligand for PD-1 in this tumor type. And then what they did, they tried to deplete macrophages using a CSF1 antibody or a receptor-targeted antibody. They did that, but what they saw very interestingly as well was that when you get rid of the macrophages, the MDSCs come back in force and they take over. And they further showed that you need to target, using this nice little diagram here, you need to target both the TAMs as well as the MDSCs to get PD-1 blockade to work. So again, thinking about this idea of cells cross-talking with one another is so important. The mechanism was even more interesting. They showed that cancer-associated fibroblasts in these tumors secreted this chemokine, CXCL2, and this was actually the reason that the MDSCs would flood into these tumors and prevent the immune response. So multiple groups now are showing a relevance for MDSCs, and certainly there are others out there. I don't have time to go through all the literature, so I apologize to those groups, but this is certainly a dominant force that is mediating immune suppression in the disease. And as part of this paper, we worked with our pathology colleagues, Dr. Krasinskis, Shishir Maithel in Surg Onc, and then two residents who worked sort of between my lab and Shishir's clinical mentorship, Mohammed Zaidi and Michael Turgeon. We were able to get resection specimens from patients with cholangiocarcinoma, create tumor tissue microarrays on a slide, and then we could stain these slides with antibodies that will recognize either the cytokines alone or phenotypically defined markers from a paper out of the Gabrilovich group that indicates their MDSCs. So these are our patient characteristics. There's some intra and extrahepatic, one gallbladder patient in the group. This is all published, so you can look at it. But this is essentially what the staining looks like under the fluorescent microscope. These are our MDSC markers, CD33 and S100A9, two of the only markers that are well characterized in formalin-fixed paraffin-embedded archival tissues that we can use, along with a DAPI stain for the nucleus. And when we merge these together, you get a nice yellow co-localization that you can see these cells present in the tissue. Now we can stain for cytokines as well in these tissues. And what you see here are co-localization, again in yellow, for IL-6 and GM, two of the cytokines we identified from our cell lines. And if you look in the actual bile duct areas of the tissue shown by the arrows, you can see this co-localization occurs very nicely. So these are present. When we quantified all of these things in the tissue, we saw a really strong correlation. The R value was actually 0.7 when you compare IL-6 and GM to one another. So those tumors that make a bunch of IL-6 also make a bunch of GM and vice versa. And again, these areas were really localized around those tumor-like regions as determined with our pathology collaborators. The other thing that had a relationship was each cytokine individually with these phenotypically defined MDSCs. So when you had high IL-6, you had more likelihood to have higher MDSCs in those same tumors, and same goes for GM-CSF. So these are very interesting relationships, albeit in a population of resectable cholangiocarcinoma. So our sample set admittedly was very biased, but for those who work in this space, you know it's very difficult to get a large number of metastatic tissue because these are typically non-operable cases. When we looked at other features of the path reports and data available from these patients to us, we saw that higher levels of IL-6, GM, and MDSC stains were all present within those tumors from patients who had satellite lesions. And then, you know, we were able to do a little bit, this is a relatively small sample size, but some preliminary evidence of factors correlating with clinically relevant metrics. We saw that in the extrahepatic tumors, patients with higher GM above the median had worse overall recurrence-free survival following surgery. So all these features indicate these cytokines and cells are present with more aggressive disease in these resectable tumors. So we've done a bit of work in showing the cytokines come from the cancer cells. That's certainly true, but one of the things we're obviously very aware of, and this is the direction we are moving to, is this concept that cancer-associated fibroblasts or stellate cells also provide an abundant source of cytokines. So in the liver, as well as in the pancreas, in many GI organs, these stellate cells or fibroblasts, they have a real job to do in a regular physiologic system. So they are typically quiescent. They can store vitamin A lipid droplets, and then as a result, they can play a role in remodeling the tissue in response to injury. So these cells become activated when there's an inflammatory event or an insult. And as a result, they sort of take action. They will go and then produce high amounts of these extracellular matrix proteins, collagen, cytokines, all sorts of things in an effort to draw in other cells and repair that tissue. So there's a bit of literature out there, of course, and much more if you take cancers in general, asking the question, how do cancer cells and fibroblasts interact to have this reciprocal, healthy relationship with one another, fostering the cancer? One of the other things that we're particularly keen on and aware of is the idea that cancer-associated fibroblasts can secrete factors that may contribute to tumor immune evasion. And this is one of the things that we've done much more work in the setting of pancreatic cancer, but we're now diving into cholangiocarcinoma as well. And just to provide context, one example of that from a paper we published a few years back in Gut, this was work done largely by my postdoc at the time, Tom Mace, who's now faculty at Ohio State. We were able to show in collaboration with surgeons that we can get primary pancreatic tumors, we can isolate the stellate cells or fibroblasts, and we can characterize them both at the gene expression and protein level to see what they're making. And you get a series of heat maps, something like this, where red is up and green is down. If we compare cancer-associated fibroblasts to normal fibroblasts, you see a number of genes are very highly upregulated. And among those, the most elevated cytokine in the mix at the gene expression level was IL-6. So that was very interesting to us. And then certainly we have a number of new targets that are abundant that we're also exploring across these disease settings. So following on these data, we were able to show in a variety, I think 4 separate preclinical pancreatic cancer models, that the therapeutic combination blocking IL-6 and PD-1 signaling had efficacy and did so in a CD8 T cell dependent manner in these mouse models. And now we fortunately have a clinical trial that's actively accruing in patients with pancreatic cancer at our institution testing this combination. After this publication, fortunately, a number of other groups published similar results with the same combination in glioblastoma, in melanoma, and in colon cancer. So it's always very nice when other groups are able to reproduce your findings in different systems using similar approaches, and kind of lends rigor and validation to what you're trying to do. And it's not a one-time we got lucky. This is clearly a phenomenon that's occurring. So we've gone back to our patient samples, and these are in blue, survey of plasma from patients with metastatic cholangiocarcinoma now. And we've shown that compared to normal donors, this cytokine is absolutely through the roof, in some cases up to 1,000 picograms per mil, circulating in the patient plasma with metastatic disease. So there's further evidence that this cytokine, in addition to being secreted from cell lines and fibroblasts, it's elevated systemically in cholangiocarcinoma as well, and other groups have published similar findings. So our group, and led by my partner in crime, Dr. Basil L. Reyes here at Emory, we're in the final stages of getting launched a combination therapy trial looking at blocking these pathways in patients with cholangiocarcinoma. So stay tuned for this. We'll hopefully be online relatively recently or soon. So just to conclude this second part, we've shown that these cytokine factors and likely others derived from cholangiocarcinoma cells can facilitate suppressive MDSCs in vivo or in vitro. We've shown relevance in vivo from resectable patients, And then finally, you know, these immune features of cholangiocarcinoma need to be continually investigated, both preclinically and in human clinical trials, so we can essentially make sense of this all. Excuse me. So to finish out here, the third part of our talk will focus on a concept that's a little more clinically mature, targeting this small molecule inhibitor against the MAP kinase signaling pathway with immune checkpoint blockade in this disease. And the way, you know, at least initially for the time being, we were thinking about this is as we see here. We have the cholangiocarcinoma tumors. They're these multicellular beasts full of fibroblasts, immune cells, and cancer. They secrete abundant cytokine signals that feed back on the myeloid and other T cell components. And then collectively, all these things feed back and limit the immune recognition of the tumors. So we think that by introducing a MEK inhibitor with an immune checkpoint blockade, you may be able to interfere with some of these processes. And the first thing we're looking at is whether MEK inhibitors directly against the cholangiocarcinoma cells can inhibit or change this profile of cytokine and chemokine secretion. The second thing is asking the question whether MEK inhibitors directly act on the T cells themselves to modulate their trafficking. There was a group, I'll show an excerpt in a few slides here, that indicated MEK inhibitors indeed can increase T cell infiltration into tumors, although we would argue that the mechanisms by which that occurs have really not been worked out yet. There's an open book for discovery. And then finally, there's some data out there, albeit a little limited, that MEK inhibitors might influence the expansion of the myeloid cells, and we're also looking into that. So we've looked in human cholangiocarcinoma cell lines. This is the signaling pathway, the MAP kinase pathway. MEK is right here. It's downstream of RAS and RAF, and then leads to phosphorylation of ERK. So if you are looking at constitutive activation, we typically use this as a target, which you can see on the Western blots here, that MAP kinase signaling is highly active across multiple human cholangiocarcinoma cell lines. And we're interested in applying this drug cobimetinib that inhibits this pathway in the cancer cells and likely other cells in the body. So the concept for using MEK inhibitors is, of course, founded on history. There is single agent data in this, this disease with MEK inhibitors. And what you can see is, you can see that in early phase 1 clinical trial, there were a few objective responses, but 43% of these patients had stable disease. So there was some modest activity just giving MEK alone as a therapeutic strategy. The other thing that was really interesting was this idea that there was a trend toward perhaps reversing the cancer cachexia syndrome, or the systemic weight loss and muscle and fat loss that occurs in patients with these advanced GI tumors. In this study, they saw increases in both muscle mass and adipose tissue when they took non-fluid weight gain into account across all patients. And our group had a collaborative paper a few years back looking in preclinical models, indeed showing that MEK inhibitors may actually act directly to inhibit some of this muscle wasting and fat wasting activity. So a second phase 2 clinical trial with this time selumetinib, formerly known as AZD6244. Patients had, again, a similar few responses and a good proportion of stable disease. What we also noticed was that, again, looking by a fancy CT imaging, a lumbar cross-section of the muscle, 84% of the patients enrolled in this trial actually gained muscle, as indicated by this analysis. And, this led to combination studies. Dr. Azad, our collaborator, conducted another trial with pazopanib and trametinib with similar disease control rates and objective response rates. So taken together, there was some evidence that MEK inhibitors indeed had single agent activity and might be something, they're not a home run themselves, but can we build off of that and do something more? So that's where, we kind of kept going. Let's see here. There we go. So other preclinical data from the Genentech group published several years back showed, not in cholangio, but in colon cancer tumors, when you put a subcutaneous tumor in and give a MEK inhibitor after it's palpable, you get a growth delay. But perhaps more strikingly, they saw a massive infiltration of CD8 T cells in these tumors. And when they combined that with immune checkpoint blockade using PD-L1, you saw this sustained inhibition of tumor progression in these preclinical models. So this was very encouraging data and suggested that there might be some synergy, although we weren't sure at first because MEK signaling is actually relevant downstream to the T cell receptor. So what you think might happen, might not happen. You have to do the work, get the data, and then we were fortunate to involve CTEP to launch this randomized phase 2 clinical trial of atezolizumab, the PD-1 targeted antibody, with cobimetinib in patients with metastatic bile duct cancer. So this trial was really spearheaded by Nilo Azad and Mark Yarshwin at Hopkins through CTEP, and we had a lot of help in navigating through Elad Sharon at NCI, who was just an amazing collaborator in getting things forward for patients. This trial, largely due to the grunt work from Nilo and Mark and many others, especially the coordinators and all the regulatory and clinical staff, opened at 21 sites across the nation. And the accrual for this relatively rare cancer was actually completed 6 months ahead of schedule. So kudos to them. Our lab essentially was, on call, receiving blood samples around the clock at a very rapid rate to do some of these correlative analyses. So this trial was exciting. It was randomized into 2 arms, the PD-1 with the atezo alone or the combination. And then again, we have a robust series of correlative studies that Nilo and I were fortunate, along with Mark and others, to get NCI funding in the form of an R01 grant to support some of this. So this work, the early data from the trial, was presented by Dr. Yarshwin at this year's online AACR meeting. I'll just briefly go through that to get everyone on the same playing field here. But 77 of the patients of the 86 enrolled were eligible and completed 1 dose of therapy. The randomization between arms was equal, and the patient characteristics are shown here that were pretty representative. This was actually a positive trial. The trial met its primary endpoint of progression-free survival, where the combination therapy is shown in red and the single arm therapy with the atezos in blue. So there was an advancement of progression-free survival in these patients, although we would certainly like those curves to be further separated, but we feel it's something to build off of. In this pre-treated population of patients, there were some success stories. There were 2 patients, both of whom were on the combination arm, that remain on therapy as of today, more than 15 months from enrollment. And as you might imagine, we're starting to do other post-hoc analyses where they may suggest intrahepatic tumors may have particular benefit compared to the others enrolled. And the adverse events, as Dr. Yarshwin mentioned in AACR, were expected and manageable overall. So some secondary analyses, and just to show you the waterfall plots, there was a nice disease control rate that was a bit higher in the combination arm, although the study was not powered for this, versus the single arm, but nonetheless encouraging. And then, 2 of the characteristics you want to look at are the responses and MSI high tumors. These were essentially balanced between the arms, and unfortunately, neither of the patients in both arms with MSI high tumors had objective responses. So we're waiting on the overall survival data to mature, and that will be reported down the road probably relatively soon. We were fortunate to get a few biopsies from these metastatic patients, and we've started to work on the histologic analysis of the tumors to determine whether changes in the microenvironment were evident. And Mark and his team at Hopkins have stained these cells preliminarily for some of the markers, although CD8 total and CD4 T cell markers don't change. When you express that as the ratio of CD8 effectors to FOXP3 positive cells, for example, we do see a shift, and that indicates that there may be an impact occurring in rewiring the composition of these tumor microenvironments. So we're doing much more staining and much more correlative analysis to see how these factors relate with clinical outcomes and, of course, the other biomarkers. So this work is early, but we're obviously going much deeper and excited about what can result from it. Importantly, when we look at the single arm from our trial, it is relatively consistent, and I won't spend too much time on this, but with other biliary cancer studies looking at single agent PD-1 and PD-L1 treatments, our response rate was essentially quite similar, largely in these unselected populations. So only a fraction of patients responded. Arguably ours was a little lower, so we might have had a tougher population to begin with, but by and large, they were equivalent. So in the interest of time, we'll wrap up here and keep going with a few more mechanistic things. Now, as we have interest in MEK, we're asking the question and going back to the lab from the clinic to see how MEK inhibitors indeed regulate cytokines and chemokines in the setting of cholangiocarcinoma. And, a lot of this work is being done in our lab by my graduate student, Amanda Ruggieri, who has taken cholangiocarcinoma cells, treated them with cobimetinib, and then developed a nice Luminex panel to very efficiently look across different cytokines. And one of the things Amanda has shown is that after MEK inhibitor treatment, some of these immune modulatory cytokines like GM-CSF and IL-1 beta that are normally secreted from the cholangiocells go way down. Conversely, other cytokines secreted from the cholangiocarcinoma cells like CXCL10 and CCL5 that normally are not there in abundance go way up, they increase. And these cytokines, interestingly, play a role in T cell migration and other immune cells to tumors. So just a brief look at some of that data here. This is GM-CSF going down in a concentration-dependent manner, and these are physiologically relevant concentrations of MEK inhibitors. Same with IL-1 beta, but as you can see in some of the cell lines, these chemokines like CXCL10 go through the roof, implying there's a clear immune modulation occurring that's might be relevant to some of the data we're seeing. We can take those cholangiocells and validate these pathways are getting hit. This work was done by our research specialist, Jacqueline Hammonds, in collaboration with Amanda, where we have various human cholangiocells. If we apply cobimetinib, we certainly shut off the pathway. But although we're seeing these big fluctuations in cytokines, the cells are viable. So these decreases in cytokines that we're seeing are not because the cells are all dying off. They're actually stuck to the plate and they're doing just fine, implying that maybe some of the efficacy of MEK inhibitors isn't on the cancer cells themselves, but it's really due to this crosstalk on the immune system that we've been interested in. And then finally, we're taking these systems that we're looking at with human cholangiocells, the cytokine chemokine production, and then doing a number of mechanistic experiments neutralizing receptors and chemokines to prove exactly how this T cell migration might occur using various assays. So certainly from our clinical trial, we have all the patient plasma saved. We have the phenotypic data coming in on the PBMCs to examine factors on T cells, suppressive myeloid cells, and then how does MEK inhibitor combinations influence clinically relevant immune checkpoints that may be either inhibitory or arguably agonistic. Maybe we need an agonist on board to better realize the benefit of this combination. And Dr. Yarshwin at Hopkins is starting to address that question in some upcoming clinical trial concepts. So Amanda and Dr. Jessica Kylson in our laboratory, who's another surgery resident, they are looking very carefully at a huge panel of cytokines and chemokines in this patient plasma to determine whether any trends emerge between arms or with outcomes. And then finally, with this as a backdrop, we have a number of challenges we have to acknowledge in cholangio research. So really in my last 2 or 3 slides, we'll briefly talk about that. The first is that we're very cognizant that patients often present with advanced disease in this setting. And the tissues that we have and that a lot of our conclusions have been drawn from are from resectable patients. So we need to find better ways to get into that metastatic microenvironment or look at circulating tumor markers and really understand the biology of this disease and its connection with the immune system. The second thing is that in comparison to a lot of other cancer types, our tools are much more limited. There are relatively few available cell lines. There are even fewer animal models that are relevant. Certainly people are working on those, but they take a long time. And particularly those animal models that can be used in the immune competent setting where we can really look at immunotherapy. And I would argue all this emphasis on PDX models is great when you're looking at tumor-targeted agents, but we're just not there yet for immune therapies and applying PDX models. So we have to be creative and go back to immune competent settings to really understand how things are going to work. So we are working in our own group at whole exome sequencing of human biliary cancer cell lines. And this is founded on the understanding and appreciation that the idea of personalized therapy in cholangiocarcinoma, in each tumor, in each patient being their own unique genetic group is very apparent and very driven by the data. So we've done some of that. Amanda, my student, had presented some of this at the recent cholangiocarcinoma conference, where we took our panel of cell lines, we've analyzed whole exomes from them, and then we see a variety of relevant mutations, and then she went back and compared them to what we see in the limited available specimens in cBioportal, and showed that many of these cell lines indeed harbor mutations that are relevant to the biology of human patient tumors. So this is a foundation, and we may be able to use these cell lines in screening relevant drugs or compound libraries to identify better personalized therapies for cholangiocarcinoma. And then finally, with that in mind, our GI group is pursuing a number of different clinical concepts founded on this idea of personalized therapy. So Dr. Waleed Shaib, one of our medical oncologists, has designed a trial where we're really looking at the personalized level, selecting patients for a number of the following DNA repair type pathways, and then enrolling them in a clinical trial where we'll give an anti-PD-1 with a PARP inhibitor to try to capitalize on this idea that some patients may harbor tumors that have greater susceptibility to DNA damage that might further invigorate the immune response. So this is going through the channels and is almost to the finish line, and we're excited to open it in the near future as well. So to conclude this last part, we've completed a very collaborative, exciting national phase 2 clinical trial of a novel immunotherapy combination that has some activity to build off of in the setting of cholangiocarcinoma. We've also shown that we can modulate these soluble mediators, and that may be relevant to how MEK inhibitors are interfacing and acting in the setting of this disease. And hopefully we'll have a number of ongoing studies, our group and many others collaboratively across the ICRN and the nation, that will focus on further interrogating these strategically data-driven immune therapy combinations in the setting of cholangiocarcinoma. So the most important slide is here. This is the acknowledgement to all the people that it takes to do this work. My own laboratory has been working very hard and coming in amidst the pandemic in a very safely, socially distanced manner to do these things. So Amanda, Jess, and Jacqueline are really the cholangio nucleus of our team. We work closely with Dr. El Reyes and Dr. Shaib, as well as Dr. Akse on the medical oncology side. We have a number of collaborators at Emory, the Olson Lab, the Paulos Lab, and then at Mayo, Dr. Tony Saab, who I've collaborated with for years, Greg Gore, who's been a really collaborative investigator, provided us with a number of cell lines and reagents to do this work. And then finally, our collaborators at Hopkins, Nilo, Mark, and many others who have really made this idea really go forward, and we really value their collaboration and insight. And much of this wouldn't be possible without support from the NCI, Dr. Sharon and Timur, and then most importantly, the Cholangiocarcinoma Foundation, Stacy Lindsay and Melinda Bikini, who have been just amazing supporters of the research we're doing and inspiring friends to hang out with at the last meeting I attended pre-COVID, which was AHPBA, and really get to know them. And if you have time, go to their website and support their mission and participate in everything that the CCF does. They're wonderful people doing things for the right reasons. So with that, I think we have a Q&A, and I'll stop there. Thank you.
Greg, thank you so much. That was fantastic, like a tour de force of immunology and cholangiocarcinoma. We do have a couple of questions, and we have only a couple of minutes, but maybe you could speak to Malin Javle's question, asking if anyone has looked at the diversity of cholangiocarcinoma types. I assume different phenotypes. And
yeah, that's a great question. And, you know, I tried to, but maybe I didn't do as good a job alluding to it as, you know, this is really the future of the way I think therapy and diseases are going. So I think in terms of the tools we have a limited number of cell lines that are sort of anatomically driven. And that was the very reason that we tried to do some of the whole exome work on the cell lines to see how well they would mirror some of the molecular phenotypes that are clinically relevant to therapy. But I think much more work needs to be done in that direction so that we can personalize things, certainly on the patient level.
And then another question commenting on GM-CSF, that in your work, it appears immunosuppressive, but there are studies, I'm thinking of Katie Kelly's study that showed some potential benefit when giving systemic GM-CSF with PD-1 therapy. What do you think about that?
Yeah, so I think this is interesting, and this is why immunology is both fascinating and complicated. There are no clear answers. GM is one of the best things that people have used in cancer vaccines and over the years in invigorating cell migration to tumors. And if I remember correctly, some of the data they're seeing is showing responses. So the data is the data, right? And I don't think we know the answer to that until we do the work where we would deplete this or other cytokines to enhance it. Like everything else, and kind of getting back to Dr. Javle's question, is that maybe GM is more prominent in the immune suppression versus the immune stimulatory effects in some patients versus others. So this is an area where there could be more individualized immune therapies, and we need to make sense of that. Certainly not every patient has high GM. There was a range of expression. So yeah. I think these are all valid studies and they're worth doing, and you know, founded on data from the past, but we have to keep an open mind.
And so I'm going to ask a question from myself. That I was on a call a couple of weeks ago, and an immunologist who I'm sure you know well made a comment about how she doesn't believe in MDSCs as a cell type, and then all the other immunologists on the call laughed, and then they went on. And I'm just curious if you could speak to that controversy.
Yeah, so MDSCs are by definition a very heterogeneous group of cells rather than a cell type. And I would say, since they really hit the pavement at the cancer immunology meetings I go to, in, you know, since 2005 and 2006, the phenotype for these cells in both humans and mice continues to evolve and be a topic of very heated discussion at meetings. So I think there's sort of a heterogeneous cell population that's somewhere stuck between a macrophage and a dendritic cell. And that's, I guess, the non-committal way to answer that. So I think both groups are right.
There's still a bunch of questions in the chat box, but I think we've reached our 2 o'clock mark, and we want to be respectful of your time and everyone else's time. So if it's all right, we'd love to make your contact information available if anyone would like to reach out to you. You know, Greg is, I can speak to, being a fantastic collaborator. So any ideas that you have, he's a wonderful person to bounce off, and if you can snag him to work with. So, and I, again, want to just thank you for such a fantastic hour that we got with you. And I think it's a great way to launch off this series.
Thanks so much. I'm happy to answer any questions. Please feel free to email me. Thank you, Nilo, and everyone have a great afternoon.
Bye-bye.
Bye-bye.